Good evening, people. Good evening. Very good evening, Kaumada. How are you doing? I think you can hear me clearly, my students. Can you hear me clearly? We're going to discuss BioEve paper. We're going to discuss BioEve paper. Kiyadala 10, ne? BioEve 10. Yes, 10. Yes, find that paper. BioEve 10. Did you answer the paper? Lamai, did you answer the paper? No? Yes or no? Yes. Okay, we'll see people. There are few simple questions. Some of these questions. All right, let's check their answers. We are starting with the MCQs. You know the procedure, right? You know the procedure. Do you know the procedure to mark it? Lamai, do you know the procedure? You're going to mark it. I'm going to say if the answer is correct or wrong. We have first 8 questions, people, to mark. First 8 questions. Can you mark first 8 question answers? Can you mark your answers, Lamai? Can you mark your answers? Right, which of the following method can be used to for the sustainable food production? People will, I think the third one is the best answer. There is no proper answer, but anyway, even I thought of taking third one as the answer. Third is my answer, your answer, improving post-harvest method, the production of disease-resistant plants and animals, disease-resistant, the production of disease-resistant animals, production of disease-resistant plants. Not wrong, neither, right? Can be used as a sustainable food production, high-yielding animals, high-yielding plants. So anyway, people, I'll go with the third one. There is no proper answer for that. Question number 2, what is your answer, Lamai? What did you mark? Viability in coastal plants, the mangrove plants, the coastal plants or mangrove plants? I think mangrove plant. Coastal is the shallow coastal ecosystems closer to the sea. I think when we say coastal, it does not describe it properly. So how about saying mangrove plant better? So I'm taking many non-living entities have one or more characters of organism. Fifth one is better answer. Fourth one is not wrong. Fourth one is not wrong. It's good as a close answer, but resource book says it is mangrove, mangrove plants. So question number 3, I think that's a clear answer we have. So my answer is fourth and your answer is also fourth. I'm happy to see that. We learn people minute amounts. What are the elements in minute amount? What we said for the elements available in major elements, carbon, hydrogen, oxygen, nitrogen, 96% is going to be mean. What are the other minute ones, people? What we said, the elements in minute amount, we said something for that. What are those elements? Come on, tell me. Beef kufe, ne? Kokro, mage beef kufe. No coal, kokro, mage beef kufe, ne? Yes. Anyway, question number 4, people. Which property of water is not caused by hydrogen bonds? To be a good solvent, important angular shape of water molecule. Not caused by hydrogen bonds. Angular bonds. How about this one? Angular bonds are not causing hydrogen bonds. High surface tension not caused by hydrogen bonds. Expansion upon freezing, hydrogen bonds liquid at room temperature. I'm taking second one as the answer, my dear. Why did you take fifth one? Liquid at room temperature due to hydrogen bonds, neither. Right? Even the fragile hydrogen bonds are present at the liquid stage. Solid state, we can see that the fragile nature changed, but anyway, hydrogen bonds are there. So my answer is second one, Lamai. I took second. So, no call, kufi, kokro. Right. Solvent because of its polarity. Solvent because of its polarity. Solvent, where did you mark then? To be a good solvent, first one. So second question, they asked the correct statement, right? Correct, no? The fifth one. Why? Now, if I analyze the first question once again, sum of all chemical activities take place in life, ne? So wrong. All irreversible changes that occur during the lifespan, that is wrong, ne? Ability to respond to stimuli from external and internal environment. Viewability of coastal, I take it as not correct. Many non-living entities have one or more characteristics of organism, correct? Ne? But they don't have all. One or more characters can be there, but they don't have all. So living, we said that when they have all the characters simultaneously, right? So my answer is 5. Sir, it is given polarity for solvent nature in the resource book. So angular shape of water molecule. Which property of water is not caused by hydrogen bonds? Angular nature is not caused by the hydrogen bonds, right, people? Angular nature is not caused by the hydrogen bonds. That's wrong, me. Right? Due to the good solvent, correct. High surface tension, you can take. Expansion upon freezing, you can take. Liquid at room temperature, you can take. So I'll take second one as the answer. Second is the answer, right? No further arguments on that. Second is the answer. Which of the following compound contain ketose as its monomer? What is it? Ketose, very good. 5, inulin, yes. Ketone, no? Lipids contain carbon, hydrogen, oxygen only, heterogeneous group of hydrophobic molecules. Carbon, hydrogen, oxygen only. Phosphate, phospholipids. What they ask? Correct or wrong? Question number 6. What they ask? Correct, I think. Eh? Only wrong. Why? Phospholipids contain glycolipids are there, phospholipids are there. Not correct. Hydrogen bonds, heterogeneous group with hydrophobic molecules. Heterogeneous group with hydrophobic molecules. Heterogeneous group with hydrophobic molecules. Hydrophobic. Heterogeneous group with hydrophobic molecules. All made up of glycerol and fatty acids every time known. Then hydrogen oxygen ratio is not 2:1 and comparatively high oxygen is present. Hydrogen oxygen ratio is not 2:1. Comparatively high oxygen present. Wrong, neither. Less oxygen. Large biological macromolecules, no. They are not macromolecules at all. So we have two. Second one is the answer. Heterogeneous group with hydrophobic molecule. Is that given? I think it's given in the resource book. Heterogeneous group of molecules. A heterogeneous group. They say lipids, biological diverse polymers, hydrophobic molecules, diverse group of hydrophobic molecules. That's correct. The heterogeneous is like different types. Homogeneous, heterogeneous. So I'll agree with the second one. Question number 7. There are 6,000 adenine nitrogen bases and 30,000 hydrogen bonds. What percentage cytosine is available? Did you work out this? I'm going to open. We'll stop the screen share. Do you want to continue? Okay. We'll see how to work it out, people. So this is 6,000 adenine. 6,000 adenine means thymine how many? DNA, right? Thymine 6,000. Therefore, adenine thymine hydrogen bonds 12,000. Hydrogen bond goes for that. Guanine cytosine, therefore 30,000 minus 20,000. 30,000 minus 12,000. Can you lamai? 20,000, 18,000. 18,000. 18,000. How many guanine cytosine? You have to divide this by 3 because each guanine and cytosine 3 bonds are there. So you have cytosine how many? 6,000 cytosine. 6,000 cytosine. So you have 6,000 cytosine, 6,000 guanine, 6,000 adenine, 6,000 thymine means 25%, 1/4. Answer is 3. Question number 8. What is correct regarding both NAD plus and FAD? NAD plus and FAD. Inorganic cofactor which are essential for catalytic activity of enzymes. Inorganic cofactor which are essential for the catalytic activity of enzymes. Which one? Inorganic cofactor can't be named lamai. Organic cofactor, ne? Wrong. Consists of nucleotide containing adenine and ribose. Correct. Act as an electron acceptor in oxidative phosphorylation. No. It is an electron acceptor NAD plus FAD. Oxidative phosphorylation. We didn't discuss that yet. Ne? Act as electron acceptor. Act as, wait. Act an electron acceptor. What we said, people? NAD? Oxidizing agent. So is it 20% right? Why 20%? 25%. Electron carrier. That's correct. Then act as electron acceptor in oxidative phosphorylation. Now, next one. Next one. 8. Right, check it once again. First one is wrong. Ne? Contain nucleotide containing adenine and ribose. Now, adenine present, ribose present, but then some extra materials are there. N and F, nicotinamide and flavin. So, second statement little doubtful. So, we'll leave that. Act as electron acceptor in oxidative phosphorylation. Oxidative phosphorylation, something happened in the membrane. You have not discussed this part yet, right? So, I'll teach you that soon. Oxidative phosphorylation. Act as a reducing agent in, so anyway, people, this third is wrong. Right? But thing is, you haven't done that part yet. We're going to do it soon. And then act as a reducing agent in cellular respiration. Reducing agent in cellular respiration. Wrong, Nedalamai. It's oxidizing agent. Ne? Act as electron carrier in cellular respiration. I think fifth one is a better answer. Right? We'll go for the fifth one as the answer. Right? We discuss certain things about NAD, NADP. Right? So, briefly we talked about it. We said that coenzyme electron carrier oxidizing agent during cellular respiration. Right? So we talked about it. So I'm taking the answer, fifth one as the best answer, people. But anyway, one of the statement, third statement, you cannot analyze because you have not done that part yet. So I'm erasing your answers. Question number 9. What is correct regarding microscope? Correct regarding microscope. What are you going to say? Resolving power of the microscope is 0.2 micrometer. What is the resolving power of the microscope, people? Resolution power? Correct, neither. 0.2 micrometer, that is 200 nanometers, correct. Scanning electron microscope, all electrons are reflected. Wrong, neither. Some electrons are reflected, right? Some are not. And then virus can be observed by electron microscope with the magnification of 10 to the power 5, correct. Ne? So I think third is okay. Question number 10, people. Select the incorrect statement regarding proteins in plasma membrane. What is your answer? Incorrect one they ask. Involving in cell recognition, correct. Ne? Some protein act as receptors, provide fluidic nature, wrong. Ne? Some protein act as enzymes and may appropriate shape joining exoskeletal fibers, maintain. Right, I'll go for the third one. I'll agree with you. Right, moving people to next page. Okay, people, 11 to 14. Can you mark answers? 11 to 14 answers. Question number 11. Below given are few organelles, subcellular components, and their functions. Select the correct combination. Rough endoplasmic reticulum, detoxification done by SCR, ne, people. So wrong. It's done by SCR. Okay, so that's wrong. Next one. Ribosome protein synthesis in eukaryotic cells. Second is okay. Smooth endoplasmic reticulum synthesis of glycolipid and no. Glycolipid. Wrong, ne? The RER, ne, lamai. And then peroxisome hydrolysis of protein, no. Glycosome converts sugars into fatty acid, no. Fatty acids into sugar, ne? Second is the answer. Centrioles absent in cytosol, wrong. Made up of actin, no. Contain 9+2 model, no. Form spindle and aster, yes. Cell organelle found only in animal cells, found only in animal cells, no. Now, it's not present in plant cells, but that doesn't mean it's present only in animal cells. Right? Fungi, they can have. So I'm taking people fourth one as the best answer. What they have mentioned in the resource book regarding centriole and its availability. Shall we check that little bit? I think there was one hint. Centrioles are made up of cylindrical structures. Paracentrioles arranged perpendicular each other, bounded by only present only in animal cells. What it says, proteins in eukaryotic cells. Centriole found only, cell organelle found only in animal cells. Centriole composed of 9 sets of triplets. A paracentrose which is arranged perpendicular. Which are non-membrane bounded subcellular components present only in animal cells. They have taken that as a correct statement. Form spindle and aster during cell division. That is also correct. Ne? Form Produce aster and spindle cell division, correct. So I think I have to take two answers. So it is an organelle, it does not bound by a membrane. Does it say membrane model? Where it is? Contain 9 plus. An organelle, anethana issue, organelle. Yeah, it is not an organelle because it's not membrane bounded. So we can't take it as an organelle. That's correct. So centriole is present, but you can't take it as an organelle, right? Why? Because this is not membrane bounded structure. So we'll go with the fourth one as the best, right? We'll go with the fourth one as the answer. Very good. Students are suggesting me, sir, centrioles not an organelle, right? Yeah, that's correct. I forgot that. So you can't say it's organelle, right? It's a subcellular structure, right, people? Which is correct regarding central vacuole of plant cell. Central vacuole of plant cell. Sodium and chloride ions dissolve in cell sap. Not sodium, we said potassium and chloride. Wrong, neither. And then the contain anthocyanin like water insoluble pigments. No, water soluble, right, people? Then surrounded by tonoplast, which is not similar to plasma membrane. Central vacuole surrounded by tonoplast, not similar, wrong. Structurally similar, right, people? Involving in osmoregulation by maintaining water balance. Involve in osmoregulation. Involve in osmoregulation by maintaining water balance. Involve in osmoregulation by maintaining water balance. Correct. Fluid-filled structure surrounded by tonoblast, which is similar in composition cytosol, wrong. Ne? So, fourth one is okay. Fourth is okay. Right. Right. Next one, question number 14 and 15. 14 and 15. Question 14, which of the following step occurs during metaphase of meiosis? You didn't learn that yet, ne? So, shall we skip that? Meiosis you didn't learn yet. So, I'll skip this. I'll skip that. Right, next one, question number 15. Correct statement regarding extracellular matrix of an animal cell. Protective layer. So, what is osmoregulation? Osmoregulation is the controlling of water and iron content present in a cell. In other words, you can see that it is basically controlling concentration inside the cell. All right. Correct statement regarding extracellular matrix. Provide layer on cell surface. Protective layer on cell surface. Join with cytoskeleton. Not affected for cell behavior, no affecting. Collagen fibers attached to integral protein of the plasma membrane by fibronectin. Correct. Major compound of elastin. Wrong, nail am I? What is major component? Collagen. So ABD. ABD. Right, we are moving to next part. Let's discuss people the structured part now. Write an example for nitrogen containing carbohydrate. Nitrogen containing carbohydrate. What are you going to write? Nitrogen containing carbohydrate. Give me one example of a nitrogen containing carbohydrate, guys. Chitin. Chitin. Cotin. Chitin. What are the monomer? What is monomer? Glucosamine. Function component of fungal cell wall. Component of fungal cell wall. What else you can write? Component of exoskeleton of arthropods. Exoskeleton of arthropods. And exoskeleton of arthropods. Right, next one. Draw how to form peptide bond. Now, how do you show that the peptide bond formation here? I'll use the same molecule N2, H2N, C, H, double bond oxygen. This is the peptide bond. N, H, CH, CH3, C, double O, OH. This one you label as the peptide bond. Right, you can give marks, people. Here 1 mark, 2 marks, 1 mark. Then I'm moving to the next one, people. Here label this diagram. What are these parts? So it doesn't matter if the hydrogen attached to nitrogen is marked down. Yeah, it's okay. Doesn't matter. Yeah, no problem at all. So what are parts, people? A, eyepiece. Function of eyepiece. Enlarged image obtained from objective lens. Enlarged image obtained from objective lens. Right. B, coarse focus control. Coarse focus control. What are you going to write? Rough focus of image. Rough focus of image by changing distance. By changing distance between specimen and lens. And lens. C. What is that? Fine focus control. Fine focus control. What do you do? The fine focus of image by changing distance between specimen and lens. Next one, D, objective lens. Objective lens. Then obtain enlarged image of the specimen. Then E, stage. What are you going to write? Hold specimen slides. Hold specimen slide. To hold specimen slide. So marks allocation, what are you going to do? Shall you give 10 marks for this? 10 marks, not bad, ne? Then what is meant by eukaryotic cell cycle? Events that occur from one division to the next. Yeah, the sequence of events that occur from one cell division to next. I'm giving 2 marks for writing that. Then state the occurrence of that take place in G1, G2 of the cell cycle. What are you going to write, people? G1, production of cellular organelles. Production of cell organelles. What else? Protein synthesis. Protein synthesis. Then S phase, what are you going to write, people? DNA replication. You can take growth also. DNA replication. What else? Histone protein. Histone protein. Synthesis of histone protein. Then G2 phase, people, cell growth. Protein synthesis. Then duplication of centrosomes. Duplication of centrosomes. So those are the events that happen here. Yeah, production of cellular organelles. Formation of cell organelles too. Correct. I'm moving to next page. Lamai, we are moving to next page now. What percentage of the cell cycle involved in interphase? 90%. Next one. What is mitosis and how it differ from mitotic phase? Mitosis is nuclear division. Mitosis is a nuclear division. And mitotic phase is nuclear division and cytokinesis together. State 5 stages. Prophase, prometa, metaphase, all right. marks අපි මේකට දාමු ළමයි 1 then ලකුණක් දාන්න 1 mark. This is 1 mark. 1 mark. 1 mark. We'll take 5 here. What is the difference between nuclear division and cytokinesis? Is it that the cytokinesis is cell division? No. Cytokinesis is the separation of cytoplasm. Separation of cytoplasm. So once a nuclear division occur, next the cytoplasmic division take place. Together, what you call the M pace, right? So together, so if you take the nuclear division together with the cytoplasmic division, we call as a cell division. We discussed that in the class, right? Nuclear division together with the dividing of the cytoplasm, we call as the cell division. Right? State events that happen in STU. What is S, people? Probably this is G1. This period is going to be the S pace. Why? Amount of DNA increase. This is G2 and what do you call M pace. This is with the cytokinesis. Cytokinesis here. So state events that happen in STU of the cell. STU. So S. What happened, people? Those things you can write once again. What are the protein synthesis, increase of cell organelles, and all. Increase of cell organelles. Protein synthesis. Protein synthesis. Right. Then T. What happened during the T phase, people? Events that occur during T. T is what is that? M pace, neither. G2 and M pace. So what happened there? T. Dividing of the nucleus, right? Replication of centriose. Nuclear division. Nuclear division resulting in two daughter nuclei. Nuclear division. Then we will U. Right? Then U. Now, people, once the cytokinesis occur, this is cytokinesis, right, people? So with cytokinesis, what happened now? The cell DNA content come down. They have given here cell DNA. Look at this. Amount of DNA of a cell, not inside the nucleus. Right now, once a nuclear division occur, people, if the DNA content of the nucleus, if they have given not cell nucleus, then people, this is going to be the anapase. This is going to be the anapase. It's coming down, people, during the anapase with the telopase. But they have not given the nucleus, they have given the cell. So cell DNA come down, people, with cytokinesis. So this phase is not anapase, this is cytokinesis. Right? So what happened here in pace U? U is interpace. So what happened in the interpace? G1, neither. G1. It's again S, isn't it, people? U and S same, ne? Because this is also G1. So you can write the increase of cell organelles, growth, neither, those events. Protein synthesis, etc. I think this is clear for you. Sir, are the G1 phase and G2 phase somewhat similar? Yeah, correct. somewhat similar, but it's not identical. Ne? Because at the G1 stage, you can see that the cell is having only, what do you call, the before DNA replication. So does 70S ribosomes in the prokaryotic cell synthesize protein? Yeah, correct. They can do. That is why they are present, right, to synthesize protein. They do that inside the mitochondria. They do that inside the chloroplast. So how G1 comes again? But it's a cycle, isn't it? Look at that cell. This is the G1 phase of a cell. Cell, take the cell cycle. Cyclic event. Take this cyclic event. You learn this cycle, right? So what is the event? This one is G1, then S, then G2, then M, C. So this is cell somewhere here, G1. This is S. This is G2 and M. So therefore, again, this has to be G1. Got it? So is tertiary or quaternary structure of protein is functional? Yes. So during nuclear division, is the chromosome number 92? Human? Human, you're asking. Right? We don't say chromosome number. You know, people, when the DNA replicate, until they separate, unless they separate from one another, you don't call them as chromosomes. So DNA content double, but chromosome number does not reach to 92. Because we take a convention, what is that? Chromosome, when two sister chromosomes are holding two, right, DNA with one another, we don't take them, count them as two chromosomes. We count them as one chromosome. So therefore, one chromatid can make one chromosome. Two sister chromosomes together can make one chromosome. We discussed this in the class. Please check that video. Right, people, now we are moving to the essay question. Briefly describe the nuclear division of plant cell. Right, let's check that one. Right, now people, this is one essay question. This is one essay question. It says briefly describe how the nuclear division take place in plant cell during asexual reproduction. You have to say that during asexual reproduction, right? During asexual reproduction, mitosis occur, right? Then you have to say that mitosis occur during asexual reproduction. Then mitosis is a nuclear division where the single nucleus divide and form two daughter nuclei, which are genetically identical. Then it has to undergo several phases: prophase, prometaphase, metaphase, anaphase, and telophase. Then you have to describe people prophase its event. Now you know that, right? What are prophase events, people? Nucleolus disappear, right? Nucleolus disappear. Sir, cannot mention centrosomes in asexual. Only. We can't say that. Have I mentioned that? If centrosome is given, sir, we cannot mention centrosomes. Yeah, we can't say. Plant cell, is that given? Then I'm wrong. Let me check. Make a lian batama. Yeah, you are correct. You are correct. Right? We can't take that. I just took one marking scheme. We can mention, yeah, we can't mention Asta also. So these two steps we have to omit, correct? Right? So let me take that. I'll erase this. So chromatin fibers, chromosomes, nucleolus disappear, chromosome arms, cohesion. Then prometaphase, the nuclear chromosomes, special protein kinetochore, and then centrosomes of each pole. That also I have to remember. right? So we have to remove people. We have to remove regarding the centrosomes and aster details. So how do they move the metaphase plate and separate without microtubules? Some more there, ne? We have to mention people the what is this question about nuclear division, ne? The nuclear division. So you don't have to write regarding the cell plate formation, right? I'll remove this point. Plant cells cell plate formation occur cytoplasm of the daughter cells. I think writing up to this point is enough. Why? Because this question is regarding nuclear division. So you don't have to write regarding the rest of the details. So the 23rd statement should be removed. Ne? No, this is not necessary. Ne? Spindle is there. Spindle present, but there is no centrioles involving in forming the spindle. Spindle present as usual, right? Only thing is the spindle organizing part is not there. Okay. So that's the answer for the question, Lamai. Right, that's the answer. So that's all for the day also. So thank you very much for joining. I'll meet you in the regular class, right? Thank you. Vanakkam. See you. Bye.